Hi there. This video is going to walk you through some helpful information to get you ready for your Raman spectroscopy lab. We'll start off with some of the basic theory behind Raman spectroscopy, and then we'll finish up with a little bit of information about the experiment you'll be conducting itself. The basic premise of Raman spectroscopy is that it is another type of vibrational spectroscopy. And you have an incident photon of laser light that you send in that interacts with a molecule in your sample, and then visible wave light gets scattered by the molecule that you detect. The key word here is that this is a scattering process. Okay, so again, notice all of these references to the scattering of light. So this is distinct from what we're used to in IR spectroscopy, where we have an absorption process where your molecule actually absorbs the wavelength of light that you send into it if it matches up with a vibrational frequency of your molecule. Here, again, sending in light, and then you're going to detect light that gets scattered in various ways by your molecule. There are three ways that your light can interact with your molecule. So the incident photon can either hit your molecule and then bounce off or scatter off at exactly the same frequency as the light that you sent in, and that's your Rayleigh scattering process, where the wavelength of scattering is equal to the wavelength of your input laser. You could also have what we call blue shifted scattering, where the light that you shine onto your molecule gets scattered, and the scattered light that you detect is going to be at a higher energy than the light that you sent in. So the wavelength of the scattered light is going to be smaller than the wavelength of the laser light. So remember that inverse relationship between wavelength and frequency and energy. Okay, so blue shifting means we're shifting to higher energies. The other process that you can have is to shift to lower energies. Okay, that's what we call our red shift. Okay, shifting to lower energies. So if you shine your light in to your molecule, and the light that you detect that gets scattered off comes off at a lower energy or a longer wavelength. Okay, so these are the three options for what you might detect in a Raman experiment. Okay, you've got your Rayleigh scattering, that's your elastic scattering, where all of the energy gets transferred by your molecule. You have anti-Stokes, where you get that higher energy scattering, and then your Stokes scattering, which is the lower energy scattering. The various shifts in wavelength that you observe by your molecule are going to depend on the chemical structure of your molecule. Okay, so similar to how you get specific frequencies out of an IR spectrum, where you have different frequencies of vibration for your CH stretches or your CO stretches, you're going to have a similar kind of chemical dependency on the frequencies of light that you're going to detect in your Raman spectra. Although we are still examining molecular vibrations with this type of spectroscopy, we do consider it to be a complementary technique to IR, so it's not a replacement to IR spectroscopy at all. We use them both to obtain a full collection of information about our molecules. Okay, and when you hopefully go on to take inorganic chemistry, you'll notice when you talk about your character tables in there, you'll see labels like IR and Raman active. Okay, and it turns out that your IR and Raman frequencies that you detect will be identical if you have a vibrational transition that is both IR and Raman active. And just one more kind of bar trivia for you. C.V. Raman, the discoverer of this type of spectroscopy, was awarded the 1930 Nobel Prize for this work, and he was the first person of color to receive the Nobel Prize in the sciences. Okay, so let's take a look at the basics of how we would collect a Raman spectrum. So the first step is to irradiate your sample with a powerful light source. It turns out that you need a really high-powered light source like a laser in order to generate a significant amount of signal that you're able to detect in a meaningful way. Okay, so see your reading in Skoog for some additional information about that. You also will measure your spectrum usually at some angle from your incidence input. Okay, so usually we detect at about 90 degrees. Again, that just kind of limits the overwhelming signal coming from your input laser light. Okay, and then the other thing that you will do is to choose an excitation wavelength that does not sit at one of the absorption frequencies for your molecule. Okay, so this spectroscopy involves what we call virtual states. Okay, and so that is again distinct from our IR spectroscopy. We'll talk about those virtual states more in a minute, but for now, just know that when you collect a Raman spectrum, you're getting information about the vibrational transitions in your molecule, similar to how you get in your IR spectroscopy. So here is a typical Raman spectrum. Okay, your laser light would be detected over here. So if we didn't have a filter, see here, there's a filter right there. If we didn't have that filter in there, we'd see a big giant peak here corresponding to the scattering from our laser light. That would be our Rayleigh scattering. Okay, and then this is just showing our one arm of our other types of inelastic scattering. Okay, so here you can see we get peaks that show up that correspond to the different types of vibrations in our molecule. Okay, so we might have a peak corresponding to an asymmetric stretch or a symmetric stretch or a bend. Okay, again, very similar to our IR spectroscopy. The biggest difference you'll notice in your spectra that you obtain with Raman versus IR is that your x-axis is a bit different here. Okay, so notice that instead of just signaling the wave numbers, okay, like you have in an IR spectrum, which represents the absolute energy of that transition, what you're plotting in a Raman spectrum is the shift, the Raman shift in wave numbers. So that shift is the shift from your incident light. Okay, so this is essentially giving you a measurement of how much of your incident light has been scattered. Okay, are you gaining energy? Are you losing energy? And by how much? Okay, that's what you're reporting on your x-axis here. Okay, let's take a closer look at the three types of scattering that might occur in our experiment. Let's take a look at our energy level diagram to start with. So again, just to point out the difference between our normal vibrational energy states that we've been discussing in class that we calculate from our quantum mechanical models. Okay, this is our traditional IR absorption that we've been discussing, where we have a transition from a lower energy state to a higher energy state, and that energy represents the difference between those two states. So if you put in enough energy of light from your laser to excite that transition, okay, then that will happen. Okay, so these are our traditional vibrational energy states. For Raman scattering, we have, remember, those virtual energy states. So the transitions are not happening within these molecular vibrational energy states like they do for IR spectroscopy. They're happening from these vibrational energy states of our molecule to what we call these virtual energy states. These are very short-lived states that just don't correspond to the absorption bands like we have down here for the molecule. Okay, so let's look at our first scattering option. So we have Rayleigh scattering. Okay, so remember this is where we shine in our laser light. So the laser light in all of these examples is going to be depicted by the blue up arrow. So we're shining in our laser light frequency, and then we're scattering out exactly that same frequency. So we see our two arrows are the same length. Those frequencies are going to be equal, so that is where we emit a photon of the same energy as our input light. Okay, so remember this HEX is our laser. Okay, so whenever we see this notation, that's your laser. That Rayleigh scattering is going to show up in your spectrum as a giant peak right at the center. Okay, so here on your Raman spectrum, this would be centered at 0. Remember, we measure the shift on our x-axis, the shift away from our incident light. So for Rayleigh scattering, we have a shift of 0, right? We're getting exactly the same frequency of light out. So we've got a giant peak there centered at 0 on our spectrum. Okay, moving on to our Stokes scattering option. Okay, so the Stokes scattering is where our molecule absorbs a photon of energy from our laser and then re-emits a photon of energy that is reduced in energy. So see that subtraction sign there. So we're coming out at a lower energy for the Stokes. Okay, so if we look at our energy level diagram, again, we have our input from the laser, which is our blue line, and then the scattering that we detect, we see is coming out at a lower energy. That arrow is smaller. Okay, so we're going to get a lower energy or a lower frequency of scattering. So those Stokes scattering signals are going to come out again at lower frequencies or higher wavelengths. Okay, so those are on this right-hand side of this particular figure, the way that it's oriented. Okay, so these are our Stokes lines coming out. And last but not least, we have the anti-Stokes option. So again, shining in our laser light of that certain frequency, and we see that we get out a longer arrow. Okay, we're going to be scattering light at a higher energy than what was put into our molecule. Okay, so this is the case where we have a molecule in a vibrationally excited state. Okay, so we're not in the ground state anymore here. And that molecule can scatter radiation at an energy that is larger than what we directed onto our sample. Okay, so again, notice the difference here. We're starting at an excited state. These options all start at our ground state. Okay, so here we're exciting an excited state molecule, and then we're detecting the scattering where it drops back down to the ground state. So we actually are scattering higher energy light. We get a larger arrow. Okay, so we get a higher frequency that's going to correspond to these anti-Stokes peaks over here. Okay, so just to get some of our terminology situated here, so remember that when you don't have a change in your energy with that scattering, that's going to be classified as an elastic scattering process. Okay, and then if you are getting a change in your frequencies and your energies, those would be classified as our inelastic scattering. So these are the same types of terminology that you encountered in physics when you talk about elastic and inelastic scattering or collisions. Okay, this is kind of like your billiard balls kind of analogy. Okay, that's what's going on here with our light interactions and scattering. practically, when you do one of these Raman experiments, you're going to filter out your Rayleigh peak. Okay, you're going to filter out that giant center peak. Okay, so normally you would not observe this. That's where our filter comes in. Okay, and that's just because that strong signal from your Rayleigh scattering will really overwhelm the smaller signals from your Stokes or anti-Stokes coming through. Okay, sometimes you'll want that Rayleigh peak. Okay, there are people who do Rayleigh scattering spectroscopy, so that's a separate kind of spectroscopy activity that we're not going to do. Okay, so we're going to filter that out, and we want to focus on we're actually going to focus on our Stokes peaks. Our particular spectrometer actually only provides the Stokes side of our spectrum, and that's okay because you'll notice that all of these peaks are very symmetrical. Okay, so the positioning of your shift on your Stokes side is going to be identical, just opposite in sign from your anti-Stokes. So you're not really getting any extra information by including the anti-Stokes in terms of your chemical shift. Okay, you can do some additional work looking at the relative ratios of those peaks. We're not going to do that in our experiment, but again, you would just use a different type of spectrometer, a different type of detector if you are in need of that information. Okay, last question I have regarding our Raman spectroscopy is you'll notice that the shifts on each side of your Rayleigh line, as we just said, are identical. Okay, but could you explain to me why the Stokes lines are more intense based on what we've just talked about in our energy level diagram? Pause the video and take a minute to think about if you can rationalize why these peaks are more intense than the anti-Stokes. And don't forget about everything you know about your Boltzmann distributions, okay, and your population distributions. Okay, hopefully you actually took me up on that offer to pause the video and think about that for a minute, and hopefully you recognize that for your anti-Stokes transitions, you're starting at this excited vibrational level. And based on what we've already discussed for our Boltzmann population distribution, right, we know that that excited energy level is much less populated under normal conditions than our ground state. Okay, so the total number of molecules that are even in this state to get excited are much fewer than the molecules that we have in our ground state available to be excited in our Stokes. Okay, so it's really just a matter of the populations of our starting points here. So that's why our Stokes is much more intense than the anti-Stokes spectrum. Okay, last but not least in our theoretical discussion is the math. We can't get away from not looking at the math at all. We'll just look at some of the highlights from all of these equations that we have presented here. So first off is recognizing that the mathematical expression for the electric field from our laser is given by this equation. Okay, that E0 is essentially just the amplitude of your electric field wave. Okay, remember you're getting this oscillatory electric field that you're familiar with from any of your discussions on electromagnetic radiation that you've had in the past. And then remember that nu EX, right, is just your, like we've seen before, that's the laser frequency of your laser light that you're using. Okay, so once our electric field from our laser interacts with a molecule in our sample, it's going to induce a dipole moment, which we represent by the symbol mu, okay, in our bond. So mu is further related to the molecular polarizability, alpha, okay, as it couples with our electric field. Okay, so alpha is the molecular polarizability, which, if you remember from Gen Chem, is really just a measure of how easily you can deform a bond. Okay, so it's a little bit more of a flexible parameter than we would have for looking for a permanent dipole moment per se. Okay, so we can a little bit more easily deform our bonds even if we don't have a permanent dipole moment. So that molecular polarizability is going to vary as a function of the distance between our nuclei, so it's going to change when our molecule vibrates. Okay, so that's described by this equation here, so we can see the dependence on essentially the bond length of our molecule and how that polarizability is changing as our internuclear distance changes. Okay, so we can, again, coupling everything together with our expressions for the dipole moment, we can get a final expression that brings everything together right here. This is not the most intuitive equation to look at. Okay, we can rearrange it a little bit using a trig identity. Okay, and once we do that, we get a slightly more manageable expression that we'll walk through. Now we'll hit the highlights of what that expression tells you. Okay, I know this looks a little bit overwhelming in its form, but we can easily pull out the information that we want about our scattering processes that are happening in our experiment with this equation. Okay, the key is to look at the frequencies of each term. Oh, and there's a missing positive sign there, so this is a third term. Okay, so let's zero in just on those frequencies that show up in our electric field. Okay, so these are all the scattering electric fields that we're going to be detecting. Okay, so if we're detecting an electric field, a scattered light at nu EX, what is that process going to be? That's the same as our laser light, right? So this means that this term is just going to represent our Rayleigh scattering. So that's how we can model the Rayleigh scattering, again, just dependent on the polarizability and the electric field. Okay. The second term we see has nu EX minus nu V. Okay, so nu V, this is dependent on your molecule. It will be different for each different molecule or each different bond that you're investigating. Okay, so this is what gives us our chemical identity purpose for Raman spectroscopy. Okay, so there we are detecting scattering at a lower frequency, at a lower energy. So if we go back and look at our energy level diagram, that means that that term is going to be corresponding to our lower energy. That's our Stokes. Okay, so we can label this. It's just our Stokes term. And then that leaves us with our anti-Stokes. So if we look over here at these frequencies, we see that they're increasing from our laser input. Okay, again, that's going to correspond to our larger arrow situation here, and that's our anti-Stokes term. Okay, so actually dissect this equation quite easily just by looking at your frequencies. The only other thing that we want to pay attention to in this equation is the appearance of this d alpha dr that shows up there. Okay, so these terms basically just show us what our selection rule is. Okay, so in order to get our Stokes and anti-Stokes scattering, in order to detect those, we need to have a change in our polarizability as the molecule vibrates. Okay, so that leads us to the selection rule for d alpha dr cannot equal 0, okay, which basically means you must have a change in polarizability to get Raman lines, which again is analogous to the need to have a dipole moment, change in your dipole moment with IR. Okay, this section is going to just briefly give you an overview of the experiment that you're going to conduct. So Raman spectroscopy is often used in art conservation science. So places like the Getty Museum or art institutes that have conservation science departments will tend to have this as one of their pieces of instrumentation that they use to study their artwork. Okay, you can use it to identify forgeries, maybe pigments are in your painting that did not exist at the time when the painting was theoretically supposed to have been done. Okay, so there's a lot of kind of forensic information that you can get out of these types of measurements. So you are going to be forensic conservation scientists and help to determine the identity of the pigments that were used in this painting, which is inspired by the painting called Flowers for Tacoma. How convenient. I wonder why I picked this one, by Andy Warhol. And we are going to identify those pigments using Raman spectra and surface-enhanced Raman spectra. Okay, so SIRS is detailed in your lab handout. We're not going to talk about that in detail here, so make sure you take a look at the handout to get your background on the SIRS technique. And this is the Raman spectrometer that we have in the lab. Okay, it's going to connect to your Ocean Optics software. Okay, you can either measure liquid samples in this compartment, which is not what we're going to do this time. We have solid samples coming from our painting. Okay, so if you notice this little knob here has the arrow pointed down, that means that the laser is pointing down so that you can measure solids that are underneath it. So there's a little sample area underneath there where you'll align your sample. Okay, so each group is going to be assigned 2 paints to identify. Everyone will identify paint number 1, and then you'll be assigned one of these other 3 paints to identify for your report. The biggest question that you're going to have for yourself is, when am I going to know when I need to do a SIRS experiment? Okay, so you don't have to do SIRS on every sample. Okay, it is a much more involved process. You have to synthesize nanoparticles and get your sample all prepped. Okay, so you're not going to want to do that unless you really have to. So our first question is going to be, do I really have to do SIRS? Okay, so looking at these various spectra here for the paints, we can see they're quite different. Okay, so let's first look at paint number 1. So that's our orange one. Okay, so for paint number 1, we see that there's a nice flat background here. And nice sharp peaks that are easy for us to identify. There's not a lot of noise. Okay, we can analyze the spectrum fairly readily. That is not a paint that you will need to conduct SIRS on. Okay, you can do your normal Raman spectroscopy on a spectrum that looks like that. Okay, on a sample that looks like that. These other samples, paint samples, okay, so if we look at paints 2, 3, and 4, right, we see they are giant blobs, almost featureless. Okay, there may be some sort of peaks up here, can't really distinguish them very well, okay, but it's nothing like the sharpness that we have for paint 1. Okay, those broad peaks are due to fluorescence that has been excited in your molecule. Okay, so some molecules are going to generate a fluorescence response and some are not. And so your job is going to be to think about which ones are going to be likely to generate a response. That's going to be helpful in aiding your identification of your dyes and pigments. Okay, so if you see this giant fluorescent signal, and fluorescence is the bane of every Raman spectroscopist's existence. Okay, it overwhelms any signal that you might get out of your sample. Okay, so we need to have a way to try to counteract that fluorescence. Okay, and that's where SIRS comes in. Okay, when you put your sample in contact with a metal nanoparticle, okay, you are able to do a surface-enhanced Raman spectroscopy that will quench a lot of this fluorescence that you see and allow the signal from your sample to come out of that noise. Okay, so that's why we use SIRS. So if you see a spectrum, if you put your sample in and your spectrum looks like a giant blob like one of these, okay, that's your signal that you're going to want to do SIRS on that sample. Okay, I think that's enough to get you started, and we will address any other issues or questions when we're together in lab. All right, hope that was helpful. Thanks.